record this if that's okay, because it's basically a short seminar. Right. So everybody, I mean, the examiners can stay. You can keep your cameras on, but everybody else, if cameras off and muted, and when it comes to questions, then we can all put our cameras back on and then discuss through the.
Yeah, just on that, Steve, I encourage questions from the rest of the audience as well.
Oh yeah, sure.
Because we're going to have 100% time with
later.
Jathushan later, so I'd encourage people to think of questions as we're going through, and please do to bring them, because otherwise they bring their perspectives on the work, which will be very relevant.
Do you want us to interrupt or wait till the end? We'll probably wait until the end before we ask our questions. Yeah. Right. Okay. Do you want to share screen then?
Oh, are they not sharing?
Can't see anything.
Oh, sorry.
And then use your mouse key as a pointer.
Yeah. Can you see this?
Okay, let me just see if I can. Okay, I'll just introduce you then, Jathushan, and this talk's going to be by Jathushan Baheerathan, a project done with ourselves here at the NCMH and also with Dr. Balash Badshah and Professor Peter Ellis at King's College, London, and it's about the glucoman and BG90 and their role as a soluble dietary fiber interaction with digestive enzymes, namely amylase and lipase. But anyway, okay, so over to you, Jathushan.
So I'm a part-time MRes student in Biomolecular Technology, and my project concerns the role of BG90 as a soluble dietary fiber, as well as its interaction with digestive enzymes, the lipase and amylase. So part A of my project was a review highlighting the properties of BG90, its structure and how it modulates cholesterol levels by interacting with lipase and amylase enzymes. Part B of the project was the experimental aspect, which involved me conducting experiments such as analytical ultracentrifugation, dynamic light scattering, and fluorescence recovery after photobleaching. So what is BG90? BG90 is a soluble dietary fiber which are abundantly found in food sources such as oats and barley. They can be taken as food supplements or be incorporated into the daily diet to help maintain a healthy functioning digestive system. The structure of BG90 consists of glucose monomer units, which are linked via beta-123 and beta-124 glycosidic bonds. The beta-124 glycosidic bonds are responsible for providing the linear backbone of the beta-glucan, whereas the beta-123 glycosidic bonds within the structure create slight bends in its conformation. And this prevents the beta-glucans from being packed together tightly, which enables it to retain a higher volume of water. Beta-glucans are also fermented by the gut microbiota to produce short-chain fatty acids, which possess anti-inflammatory properties, which encourages a healthy digestive system. Here we have an illustration of the structure of BG90 where we can observe the beta-123 and 124 glycosidic linkages. So the functions of BG90. BG90 is capable of reducing levels of LDL cholesterol in the blood. This is due to BG90 being able to form a viscous gel-like substance when interacting with water, which can trap bile salts. And the encapsulated bile salts will be excreted from the body, causing a decrease in bile salt levels in the body, which consequently causes the liver to absorb more LDL cholesterol to produce more bile salts to compensate for this loss, resulting in the reduction of net LDL cholesterol levels. The viscous gel-like substance formed also creates a physical barrier, which prevents the lipase enzyme from accessing lipids, which will prevent the production of fatty acids, which will in turn reduce the levels of cholesterol formed in the body. BG90 is also able to act as a non-competitive inhibitor for both the lipase and amylase enzymes, which disables the enzymes from being able to interact with their substrates. These are the results gathered from the AUC at different concentrations of BG90. The single peaks shown on the graph to the left show that the BG90 is pure and that there are no other species present in the sample. We can also see that at low concentrations of BG90, the sedimentation coefficient is higher due to the concept of non-ideality, because there are fewer particles present, therefore there will be fewer collisions and intermolecular interactions between molecules. The graph on the right shows the sedimentation coefficient being extrapolated to zero concentration, which is essentially to eliminate the effects of any hydrodynamic interactions to gain the true hydrodynamic properties of BG90. These graphs show the sedimentation and DLS profiles from the interaction between BG90 and lipase, and we can observe a small peak at around 7.5 S Swedbergs in the mixed sample, which is not present in the BG90 and lipase-only samples, which could indicate the presence of co-lipase. And from the DLS data on the right, we can observe that the green lipase peak disappears, which could indicate that the number of free lipase molecules has decreased, and lipase could be forming a larger complex with BG90. We could also observe that BG90 alone is aggregating, as shown by the red peaks on the graph. The AUC and DLS experiments were also repeated to monitor the interaction between BG90 and the amylase enzymes too. And here we can observe that on the mixed species, there seems to be a slight shift in the amylase peaks towards a higher S, which indicates that species are getting larger and so are sedimenting faster, hinting at a possible interaction between BG90 and amylase. From the DLS data, we can see that the green amylase peak seems to disappear, which could indicate very large aggregate formation, which could not be picked up from the DLS itself. But from the results gathered from these experiments, it's not entirely certain whether there is an interaction occurring between BG90 and the enzymes, which is what led us to conduct the FRAP experiments to gain further insight into the interactions that could be occurring. It's a little hard to see, but these are the experiments that these are the FRAP experiments that were conducted.
Jathushan, if you did full screen mode, you might be able to select better. Okay, that's a bit more clear. Okay.
Yeah. There was a lot of data to put into multiple different slides, so I just condensed it into one. So here, both lipase and amylase enzymes were photo bleached and tested for fluorescent recovery using a confocal microscope. And these are the results that were gathered from that analysis between BG90 and lipase at different concentrations of BG90, ranging from 0 mg per mil up until 5 mg per mil. From this, we can observe that at a higher concentration of BG90, the fluorescent recovery is noticeably slower compared to the sample which had no BG90 present, which indicates that there is an interaction occurring between BG90 and the lipase enzyme, and possible binding due to the recovery being slower and never 100% recovery, because the lipase enzyme is unable to diffuse freely back to the photo-bleached region. This experiment was also repeated with the same parameters on the amylase enzyme. And similarly to the results of the lipase, when using a higher concentration of BG90, the fluorescent recovery of the amylase enzymes was reduced due to the interactions between BG90 and amylase, preventing the amylase enzymes from being able to diffuse freely back to the photo-bleached area. And to the right, we have the results from the control variable, which consisted of just the marker. And it's from this we can see that the recovery was much faster compared to the samples treated with BG90. So to conclude, what we found was BG90 is important to incorporate into the daily diet due to its healthy benefits, such as reducing the levels of LDL cholesterol by encapsulating bile salts, forcing the liver to absorb more LDL cholesterol to produce more bile salts to compensate. Is also able to interrupt the digestion of lipids by acting as a non-competitive inhibitor, as well as forming a viscous gel-like substance to form a physical barrier between the enzymes and the substrate. The data from AUC and DLS were inconclusive, which led us to performing the FRAP experiments to gain a deeper insight into the interactions that could be occurring between VG90 and the enzymes. Yeah.
Okay, thank you. Yes, Susan. Could I ask a quick question?
Yeah.
With the FRAP measurements, I mean, to some extent, we're taking your word for it that the recovery time is slower, simply from the shape of the
from the curve.
Of the curves. We're in the presence of BG90 for both amylase and with lipase. So does the software produce some sort of recovery time parameter like TOR-H or something, which is characteristic of the recovery?
Yeah, so we used the LASX software to conduct this experiment, and what the program does is essentially we give it its parameters such as the power of the laser and how much of the photo bleaching time we give it. So we give it a time frame to bleach it for this long, and then we measure the recovery for a specific set time. And then this is the curve that it generates from that input.
Yeah. And the second question is that it looks as if the bits of evidence that this is science, you can't just rely on one technique, you have to, it's like a jigsaw puzzle, which you put all the pieces together, and then when you get several techniques, either giving complementary information or confirmatory information on the others, that's how it works. When it doesn't work, then it gets interesting. But okay, you showed there is the amylase and the lipase are both interacting with BG90. But can you say, from the information you gained so far, which is which is interacting most strongly? The lipase.
It's still really hard to tell because of how similar the graphs look to each other. But I think what we could have done if we had more time is calculate the diffusion coefficient from the data given, and from that possibly could give us a set value to tell us how much of a difference the interaction is between both enzymes.
Okay, so if there was further work, that's what you would be doing.
Thanks, Steve. Let's pass to Richard. I think he's got a next question.
I do. Jathushan, very good presentation. Thank you. So what you're saying is, are we saying there is an interaction between the two molecules?
Yeah, it seems apparent that there is some sort of interaction due to how affected the recovery is between both samples.
So I've got two questions. So first of all, can we confirm that this interaction impacts on the ability of the lipase enzyme? And my second question is, if it does, is that a competitive inhibition or a non-competitive inhibition?
I believe it to be, it could be vice versa, because I do know that it does have a tendency to bind to the allosteric site of an enzyme from previous studies I've read. But because it also has other functions such as forming a physical barrier, I feel like it also competitively prevents the substrate from being able to interact with the enzymes directly itself.
Brilliant. Okay, thank you.
Other questions? Latika, Miyang? Sure, you've got another one, Steve. We can come back to you in a sec.
It was a very good presentation. So, do can we use both the enzymes? Like, does the concentration of the enzyme affect the how they react with the substrate?
It could do, since we've only measured a specific concentration of enzyme, we only generally change the concentration of BG90 itself. So perhaps if we could try maybe various different types of concentrations of the enzyme, it could have some sort of an effect. It could maybe make the reaction a much more faster reaction.
Yeah.
Yeah.
Thank you.
Yeah, Steve?
Morning.
Yeah, I don't have question. Okay.
You don't have a question? No, okay. That means I can ask my question. Yeah. Okay. I mean, obviously, in a limited timescale, you couldn't do a matrix of different concentrations and things to see what's going on. But how does this translate to reality? So let's say that, and it seems so, that the glucomans do have a cholesterol lowering effect, which is good news for all men like me who've got to use statins and all this sort of stuff to keep cholesterol down. But realistically, how much, because don't forget these things get diluted in the blood and everything, how much, or in the gut, which is where it's important, how much glucomannan do we need to consume for it to have?
Yeah, so the
effect?
issue with that is because we only utilize porcine enzymes, it's not human enzymes, so it could react very differently compared to what we've seen in experiments. And yeah.
But that's a good answer. But what if you were like porcine?
Yeah.
Then what quantity of, I mean, what sort of foods contain glucomannan?
Oats, barley, wheat, stuff like that. Yeah.
Okay, good. So how much do we need to eat loads of
Not
wheat
loads,
mix
but if it's incorporated into the diet, I'm pretty sure it could have a noticeable effect of lowering the LDL cholesterols from your body.
Okay.
Yeah.
Okay. That's always important to sort of transfer what you see in the lab to what is reality. Okay. Thanks.
Alan, feel free to chip in now. Everyone's had a go. Richard's got his hand up. I'll pass to Richard, but
yeah.
we can
So
dive back in there.
It's a quick one. So you mentioned that the beta-glucans, they can come from oats, barley. What's the function of those polysaccharides for those plants? What does it do in the plant?
I believe because they have a property of high water retention, because of its structure, it's not really tightly packed. It can retain a high volume of water, which I think is essential for plants to do that, especially for plants living in very hot climates.
Right. Okay.
Ian, Alan.
Okay, I can ask a question. In your figure 3, you mentioned that that there was a small peak, and you say that it could suggest there was a presence of co-lipase. Where would that co-lipase come from?
Let me just get the figure. You said figure 3 from the interaction between BG90 and lipase?
Yeah, that's right. Yeah.
Yeah. So I believe it to be interacting with the BG90.
So but you've just added lipase. So where did the co-lipase come from?
And I just believed it to be.
Sorry, go on.
Yeah, I just believed it to be the BG90 interacting with lipase, forming some sort of aggregate that's being detected in the AUC and DLS.
But the co-lipase, is it already in the? Well, I think what Alan's asking is, where does the co-lipase come from? Is it in the same jar?
Yeah, it could very well be in the sample that we've used.
I mean, is that normal that you would have some co-lipase with your lipase when
No,
you
it's not
in the
normal. 'Cause
sample?
we did have trouble dissolving the solution itself. It did take overnight to do. So I'm guessing that sample wasn't really in its purest form.
Okay. So from the actually sitting in the sample,
Yeah.
from your wherever you've got your porcine lipase from, there will be a small amount of impurity as co-lipase. Is that,
Yeah.
I think that's what you're suggesting, yeah?
Yeah.
Okay. No, that's fine. I was just intrigued whether you added some or something like
no.
that. No. Okay.
Wonderful. Steve, how much longer do we have for the open questions?
That's it.
Okay, well, it feels like a natural point to pause then. Thank you for the presentation from my side. I'll pass back to Steve as you be chairing.
First of all, I'll stop recording. It'll be a bit embarrassing if. Okay, so we'll stop recording now.